Good morning, friends. Yesterday we studied the gene, its nature. Today, along with the nature, we'll study the terminating part of the topic, that is the expression and regulation also. Now here, first of all, the yesterday's bit will continue, that is protein synthesis process after the formation of genetic code by different scientists, and that genetic code was accepted worldwide, and finally this genetic code is utilized and used in variety of the ways. Here the protein synthesis, the next step, that is central dogma of the protein synthesis, or central or main theme of the protein synthesis. See, the central dogma of the protein synthesis here involved in this small reaction, that is DNA, then mRNA and protein. The whatever the sequence of the DNA get copied or transcribed, the script of the DNA get copied and written by this mRNA, and that sequence on the mRNA which get copied from the DNA, when it comes out of the nucleus to the cytoplasm of the ribosome, then it converts into protein, that is polypeptide chain is formed by translating the sequence of mRNA which is just copied from the DNA, and finally it translated in the form of amino acid to form the polypeptide chain. Finally, that is nothing but the protein molecule. That is the central theme of this process. This is the elongated, lengthy process. Now see, for the protein synthesis or formation of polypeptide chain, what are the requirements? See, the first of all, we must have the DNA to provide the base sequence, first of all. Then second, there are 20 amino acids, different amino acids are required. Then non-genetic RNA, that is mRNA, rRNA and tRNA also required. Not only that, ribosome, you know that in the cytoplasm, the ribosomes are not as a complete unit. That is, in prokaryotes, we know that there is 70S ribosome, in which the smaller unit is 30S and larger unit is 50S. But in eukaryotes, the smaller unit is 40S and larger unit is 60S to get final the A.T.S. ribosome unit. Now these two ribosome units, this smaller unit and this larger unit, these are not together in the cytoplasm. During the process, these two units get together, so that the ribosomes are also necessary, that is smaller unit with larger unit. Then various enzymes, factors for the carry on the reaction. Without the factors or without the enzymes, the reaction cannot be carried. Then energy source, ATP and GTP. This is the requirement for the protein synthesis. Now the central dogma of the protein synthesis is mainly distributed into two steps. The first step we want to study there, that is the transcription. Name itself indicating the meaning. The script is copied or copy first and then written on the mRNA. Whatever the sequence of the DNA, whatever the codons present on the DNA, whatever the nucleotides present on the DNA, that sequence get copied by the mRNA. During the process, the script of the DNA get copied on mRNA, so called as transcription. Transcription means the total sequence, complementary base sequences get copied by the mRNA during the process, that is called as transcription. The transcription units has different like the promoter, then structural genes and terminator. Out of that, the DNA which is the main role on which the sequence is present of the nucleotides, that sequence is get copied by mRNA. Now that DNA is actually the main part, that is DNA deliver the sequence of itself to the mRNA during the process. So mRNA just copying the sequence on DNA into the nucleus. The total process of the transcription is carried out into the nucleus here. Now what happened during the transcription? The two strands of the DNA, out of which the strand which is allowed to copy for mRNA, that strand is said to be the template strand. What is template strand? The template strand is that strand from which the mRNA get copied from the DNA. The template strand is that strand which is just copying the sequence of DNA, nucleotide sequence on the DNA on the mRNA. That is, template strand is in 5' to 3' direction. The remaining other strand of the DNA is nothing but the coding strand. The strand from which the mRNA not copying the sequence, that other strand which is in the direction of 3' to 5'. Now that is called as coding strand. Such two strands of the DNA, actually from the template strand, mRNA copying the nucleotide sequence and other strand is coding strand. Now the promoters, the terminators and the structural genes are essential for the process. Now promoters, what are the promoters? Promoters are nothing but the small sequence of on the DNA. It is nothing but small sequence on the DNA that is allow to the process of protein synthesis or to promote the process of protein synthesis. Okay. Now promoters are the DNA sequences which promoting the process of protein synthesis. What are the structural genes? The structural genes are nothing but the like Z, Y, A, like structural genes are present for the protein synthesis. Then the terminators are also required for the termination, the process, that is factor, initiation and termination are also necessary for the process of the transcription. Now what happened initially? The promoters, structural genes and terminators are used here for their role. The sequence of the DNA which allow to terminate the process is called as terminators. Terminators and promoters playing important role in the transcription. Initially, the strand of the DNA molecules of the DNA get separate from each other, that is using the enzyme. Two strands of the DNA get separated from each other, that is the separating due to the enzyme. The enzyme is called as the endonuclease, that is breaking the bond between two strands of the DNA, and two strands get separate from each other. Now during this, the bonds, due to breaking of bonds, the two strands having the single nucleotides hanging over. Now these nucleotides, out of that, the strand which is in the direction of 5' to 3' is taken for the copying by the mRNA. Now this mRNA copying this sequence on the DNA that stateless strand, the mRNA first of all match up the nucleotides present into the nucleoplasm. The sequence is complementary to this DNA sequence, that is on the sequence which is present on the DNA template strand, and one by one with the help of polymerase enzyme the sequence of the mRNA, that is nucleotides of the mRNA, are matched up with the complementary DNA strand according to that, and a sequence is continuously made. Now the process of starting of the matching the mRNA sequence on the template strand, the initiation takes place. The process of transcription is get started here. Now first of all, mRNA match up with the complementary sequence, nucleotide sequence present on the DNA and get copied first of all. For that, the enzyme with the RNA primer is necessary. Now enzyme with mRNA now start the initiation. The initiation factor is necessary for that, and first match up nucleotide is get copied first of all on the mRNA. Now the initiation continues into elongation. One by one, the process of complementary nitrogen bases present on the template DNA strand with the mRNA get complement, and finally the elongation of this such DNA sequence of nucleotides on mRNA also takes place, so that the process is get elongated and the sequence on mRNA get copied, the process elongated, and finally the process towards the 3' end going to terminate. For that, the terminating factor is necessary. The termination takes place. Before that, whenever the process is going towards 3' end of the DNA template strand, the mRNA also got the signal to terminate the process, so that the termination result into the enzyme which is attached to mRNA, that is remains for a while, and with the enzyme and mRNA both get finally at the termination fall down. Now this mRNA now has the copy which is in writing of the mRNA DNA template strand. Now this copy, whenever transcript from the DNA template strand to mRNA, that time the enzyme and the mRNA get fall down after the termination and get separate from this process. At the same time, afterwards, whenever the termination takes place, the mRNA which is get transcribed, which is having the transcription or script in writing of the nucleotides from the DNA template strand, comes out of the nucleus and comes into the cytoplasm through the nuclear pore. Now this cytoplasm having the ribosomes, such structures, and that their next process, next step of the central dogma, the sequence or codons present on mRNA which get copied from the DNA get translated in the form of amino acid. That is the second step of this process. Now after transcription, the translation. Transcription, the script is get transcribed from the DNA. Now mRNA comes out of the nucleus, and now this sequence on the mRNA get translated with the help of or translation result into formation of amino acid. Now all amino acids one by one get formed after the translation. The translation, which translation? That is the triplet codon present, which is copied on the mRNA, that get translated with the help of tRNA, and finally the result into formation of amino acid. Such many amino acids are linked together with the help of polypeptide chain, and this complete polypeptide chain now called as protein. Now so that mRNA get translated. This mRNA not a normal mRNA. These mRNA are just stolen the message, copied the message of the DNA and come out of the nucleus to the cytoplasm. Now the sequence in the form of triplet codon is get translated into protein or polypeptide chain. The process here involves, first of all, for this, the first step is that activation of amino acid tRNA complex. In this process, the normally tRNA are necessary, and for that the activation of amino acid and tRNA complex formation is essential process initially, so that in presence of enzyme amino acid tRNA synthetase, this enzyme provides the activation of amino acid and formation of amino acid tRNA complex. What happened? This enzyme amino acid tRNA synthetase is helping to amino acid to activate, and tRNA with a particular amino acid with anticodon already present on tRNA are ready or get activated first of all in this process of translation. The second step of the translation, that is formation of polypeptide chain, that is formation of polypeptide chain is involving two steps. That is, number one, first step is initiation. The second step, elongation, and third step, the termination. The translation continues through these steps. The first step, initiation of the translation takes place. During this step, what happened? The mRNA are having the direction, that is 5' to 3', on which triplet codon, starting codon AUG is present at 5' end, and continuous such triplet codons are present on the same mRNA, that is just copied from the DNA. The script is copied from the DNA, same mRNA now taken in for this process of translation. The initiation codon present on that mRNA is AUG, and different triplet codons are present continuously, and finally at the 3' end there is a stop codon or terminating codon or nonsense codon, that is UAA here. For this translation process, the ribosome units are not like this way, they are not attached together, they are dissociated from each other. That is, smaller unit, that is lower is a smaller unit, and upper apple-like structure is a larger unit. In prokaryotes, these two units are of different size. In prokaryotes, the smaller unit is 30s unit and larger unit is 50s unit to form total complete ribosome 70s. But in eukaryotes, the smaller unit is 40s unit and larger unit is 60s unit, together forming 80s unit, that is ribosome. But these ribosome, during when the process is going to started, at that time they are not get together, they are brought together. First of all, the larger unit smaller unit comes and join to the mRNA, binds to the mRNA. Now this smaller unit, afterwards larger unit also come together to form complete ribosome. Now this ribosome, smaller and larger unit, that is 80s unit of the ribosome, binds to the mRNA here. Now this ribosome having three sites. The P site, P for polypeptide chain, therefore it is called as P site. The A site, that is amino acid site, and this is E site, that is the process empty tRNA get exit from this side. So this is called as E as an exit site, P for polypeptidal site and A for amino acid site. Now during the process of initiation, the starting codon is present on mRNA, that is AUG, the first tRNA complex, amino acid tRNA complex, which has the methionine the methionine amino acid tRNA complex come with AUG triplet codon, and in the ribosome the first amino acid is get matched. Now only this is the condition, initial AUG, this triplet codon with amino acid the amino acid tRNA complex, only this first AA1 amino acid tRNA complex only enters through this site, P site. Remember, only amino acid first tRNA complex enters through the P site, and the remaining amino acid tRNA complexes are entered through the A site. Remember, only this is the first amino acid, which is called as met or methionine tRNA complex, is entering through the P site first of all, and first amino acid is to be get translated of the AUG as a triplet codon from the mRNA. AUG, which is the initial sequence of the process, is get translated into formation of methionine, the first amino acid, and at every time whenever the one amino acid is to be prepared, now the ribosome, this complete ribosome is moves to the next triplet code. At every time, at every place, whenever the one amino acid is to be prepared, the second time the ribosome moves towards the 3' end from 5' end. Okay, one triplet codon is moving at every time by the ribosome, and this movement of the ribosome is called as translocation. Ribosome moves every time one triplet codon ahead. Now this movement of the complete ribosome set is called as translocation. One by one amino acids are linked together. Now first amino acid prepared the link with the second amino acid. Now the link between two amino acid with the help of a polypeptide bond, that is called as peptidal bond. Now one amino acid, second amino acid, that is amino acid first, amino acid second, amino acid third, so on. Likewise, each triplet code results into formation of amino acid, and there is empty tRNA. Now after the formation of amino acid goes towards the E site and get exit from this ribosome, so that the next tRNA complex comes from this A site, join with forming the amino acid, and in this way there is a formation of chain. Now initiation of the process is followed by elongation. The chain of the amino acid going to elongate it with the help of polypeptidal chain. So continuous process is going towards the 3' end finally, and such polypeptide chain, that is amino acids, are linking together with the help of peptidal bond and forming the polypeptide, and goes towards the finally elongated, and elongation completed. After that elongation, the last step, the translation, is called as termination. When ribosome moves towards the 3' end, finally at the last time mRNA sequence triplet codon sequence is now not giving any sense to prepare the amino acid, so that UAA, such a triplet codon, is nonsense codon, which is not giving any sense, which is not resulting in the formation of any amino acid. So the process of the translation indicates, or indicator is given to stop this process, to terminate this process. So the termination takes place. UAA, this triplet codon, is used as an indicator for the process to stop or terminate the process. Now in this way the termination takes place when the process is continued up to at the end of the 3' end of the mRNA, and finally termination takes place. As a result, the chain of amino acid, all the amino acid in this process are linked with the help of peptidal bond. Now this complete set of amino acid linked with the peptidal bond is nothing but the polypeptidal chain, and that is nothing but the protein molecule. Complete set of the polypeptide chain is now called as one protein molecule. Now here the ribosome used, single ribosome is used to form a complete protein molecule. Sometimes whenever in the same process ribosomes are 2 or more than 2, and no doubt the resulting protein molecules are more than 2 then, and such a condition is called as polyribosomes or polyribosome process of formation of polypeptidal chains. Okay, now this is complete process of the translation. So that central theme, that is DNA, first of all get copied by the mRNA. mRNA comes out of the nucleus, and that triplet codon present copied on the mRNA, that is get translated into formation of protein molecule, and in this way the protein synthesis get completed here. Now this is the actually the protein synthesis. Yesterday we not completed. Here we just completed the process of protein synthesis.